everyone. Can you hear me well? I hope so.
Yes, we can hear you.
Okay, perfect. Sorry for the slight delay, but our previous lecture overran a little bit. But without further ado, it is my great pleasure to introduce our next speaker, Aakrosh Ratan. Aakrosh is an associate professor of public health sciences at the University of Virginia. His group's main focus is on cancer genomics, and with his team, Aakrosh develops and applies statistical and computational solutions to understand how mutations and other alterations can contribute to the pathogenesis of a variety of cancers. His research combines genetics and genomic techniques with computer science to develop new ways of gaining insight into genome biology. So that genome biology, in fact, is the genetic basis of traits and evolution after all. So today, for our NGSprint lecture, Aakrosh will tell us about molecular subtyping of multi-omic data using SUMO. And without further ado, I give you Aakrosh Ratan.
Can you hear me now?
Yes, now we can hear you.
Okay. All right. So, can you see my screen now?
Yes, very well. Thank you.
All right. Thank you. And, I would like to thank the organizers for this invitation to talk about some of the recent work we've been doing in molecular subtyping. So, generation of multiple data types is quickly becoming the norm in biology. Data is collected from a pool of molecules. So what you see here are circles, and they refer to molecules, and each genomic assay can be thought of as a layer. Now there are interactions between layers and within layers. The interesting thing here is that except for the germline, all other layers that you see here reflect both genetic regulation as well as environmental interactions. And that is one of the reasons why we've had this genome-first approach. The idea that the germline will serve as anchor in studies. And that has also driven a lot of the method development in this field. So let's take a look at a few examples before we go forward. So what I'm talking about is things like QTL analysis. And so what you do is essentially identify germline variants and then identify if a particular, determine if a particular germline variant can be associated with the expression of a gene, either in cis or in trans. You can also think about analyses like allele-specific expression, where you are testing whether the maternal or the paternal allele is preferentially expressed. And all of these are genome-first analyses, but they're also examples of what we call a multi-staged analysis. So the idea here is you start with a single data type, and in each step, you bring in another data type. And hopefully what you are doing is increasing the signal to noise ratio every time a new data type is added. But there are other methods of integration. For example, if we assume that every genomic assay can be represented as a matrix, right? So you have the patient or the individual in a row, and each column represents a feature. In this case, it could be a SNP, or it could be a gene in an RNA-seq experiment, or in an miRNA experiment, it could be a microRNA. If you have the data in this types, you can actually just concatenate them. So here we bind the columns of this data together, and now we assume that this data is coming from a single data type. There is an obvious advantage to this way of doing things, right? We've developed a plethora of methods that work with a single data type. And once we have made this assumption, we can apply all of those methods to our data now. The obvious downside is, well, you're just concatenating data. You in fact want to filter each data set appropriately, and hence feature selection becomes really important. Then there are methods like model-based integration, where the idea is that you perform your classification or your clustering on individual data types. So for example, if this is somatic mutations, you actually do the clustering on somatic mutations. You do the clustering on RNA-seq separately, and then you add everything together or integrate everything together. Again, the upside, we can use existing methods. The downside, in this case, is you miss out on any interactions because you are modeling each data type separately. Then there are methods that we are going to talk about today, on which SUMO, which is a method that I'm going to discuss or present today, is based on, and that is transformation-based integration. The idea here is to take each data type, transform it into another data type, and then perform the integration in that realm. So what do I mean by that? So what I mean is you could, for example, transform this SNP cross patient into patient distances, and I'm going to talk about that in a minute. And once we have done that for every data type, if we can then intermediately merge these intermediate representations, then we have an advantage because now we are integrating different types of data and we have a unifying feature within the data types. Now, some people like to use pathways that are known if you're looking at cancer data sets, right? So we know oncogenic pathways. So one idea would be if we could map every data type onto pathways, then that could be a unifying feature. Obvious upside to this is that you can handle data type specific noise, missingness, and all those data type specific properties at the level here. And then once you have converted it into an intermediate representation, you can then go on to integrate. So the transformation-based method that I'm going to talk about today is based on the idea of patient similarity. This is an effort to model how medical researchers think about patients, right? So the idea that let's look at clinical data types and think about each patient as a node in this graph. And we can think about the edge as being thicker between two patients. In other words, the edge weight is higher between two patients if they are very similar to each other, right? And if you do this for all pairs of patients, then what you will have hopefully is a cluster of patients that are very similar to each other connected by thick edges, whereas clusters or pairs of patients that are not as similar to each other will be connected by thin edges. And we can do this for every data type. And this idea is not novel, right? This idea has been, I think, the first paper that I have seen so far written in 2004. And let's take this example. This is an example of intensive care unit database. So every time a patient comes to the ICU, there are certain parameters that are collected for the patient. And this study looked at the cosine similarity between those patients. And what they found was that based on these features, if you could calculate the cosine similarity between patients, you could easily predict the outcome of the patient. And that's profound, right? I mean, that's one of the things that we want. So let's come back and think about this in terms of generalizing this idea. So let's say we have a particular data type. We can convert it into a patient similarity network. We also have other data types, and if we can convert each one of them into separate patient similarity network, then figure out a way to integrate all those networks. That would allow you to essentially perform all the machine learning tasks that we are interested in, be it supervised or unsupervised. So today I'm going to only talk about the unsupervised clustering part of this framework. We have ideas about classification, and maybe we can talk about it some other time. But today the talk will focus primarily on unsupervised learning from patient similarity. So let's break this process down. We want to take each genomic assay. We want to first figure out how to calculate similarity between two patients, right? Once we have figured that out, then we want to figure out how we are going to integrate all these representations. And once we have done that, then the question is, how are we going to cluster those into multiple groups? And there are several issues with that. How do we ensure that the resulting clusters are stable? How do we identify what is the right number of clusters and so on? So we'll talk about all of those problems briefly. All right, so let's take an example that I think more or a large number of us will be familiar with. Right? So let's take an example of gene expression, which is measured using an RNA-seq assay. What that RNA-seq assay gives you is a matrix. With the rows, you can think of representing genes, and the samples are in columns. And what you want to do is convert this information, which is an M cross N matrix with M much, much greater than N, into an affinity matrix, where the values here represent the adjacency or the similarity between patients who we are going to consider as nodes in our experiment. All right, so let's say we start our RNA-seq experiment and we get our counts. Then, similar to what you do for most expression-based analyses, you are going to throw away the genes that are not expressed in most samples, right? Once you've done that, you are going to essentially do some sort of a normalization. And the idea behind that normalization typically is that the variance in the data should not be dependent on the median, right? So you will either do, let's say, a variance stabilizing transform, or you will do a log normalization. Once you've done that, then similar to if you were doing a PCA analysis of RNA-seq data, you're going to scale the data. So you're going to make it so that the mean expression of each gene is 0 and the standard deviation is 1. Right. So now we have pre-processed expression data. The next step would be to take that pre-processed data and convert it into a distance. Now, for those of us who are familiar with RNA-seq examples, you essentially do this as a QC metric, right? Typically, if in your experiment you are doing controls versus cases, you want the controls to cluster together and the cases to cluster together. And in order to do this, the typical way to do this is to use Euclidean distance in order to calculate patient or sample distances. So we essentially apply those Euclidean distances and calculate distance between every pair of patients. And now the next step we want to take is to convert those distances into similarities. There are lots of ways of doing this. You can just scale the distances, right? What we choose to do is to use what's called a radial basis function kernel. So what you see here is the similarity between two patients, I and J, is e raised to power negative square of the Euclidean distance, and the rest of the parameters can be thought of as defining the shape of this Gaussian function. Now, instead of just using this as is, we think about it a little more in an adaptive manner. So what we have is, instead of that sigma square, we replace it with epsilon I and epsilon j, where epsilon I represents the average distance of the ith individual to its k nearest neighbors, right? So essentially, we are trying to put all the patients who are connected to patient I in terms of the distribution of distances of all the patients. And once you've done that, you now have a similarity matrix, and you have effectively converted a gene expression assay into a similarity network. You can do the same for methylation. You know, there are caveats related to how you would pre-process methylation, but you would definitely perform some sort of filtering, followed by some sort of normalization, followed by scaling, so that each probe in methylation or each loci in methylation is scaled to have 0 mean and standard deviation of 1. Once you do this for every data type of interest, you essentially have, let's say you started out with 3 different data types. So you now have 3 similarity networks, A1, A2, and A3. And for now, let's assume that you have data for all the data types for all the patients. So each one of this will be an n cross n matrix. Now, an interesting point to remember about these matrices is that these are going to be positive numbers. Remember our radial basis function. If the Euclidean distance is 0, it evaluates to 1. And if the Euclidean distance is extremely large, it evaluates to 0. What that means is the similarity matrix is always non-negative. And hence we can use what's called non-negative matrix factorization, where the idea is to decompose each one of these matrices into H, S, H transpose. S is assay specific, so A1 will decompose into HS1 H transpose, whereas A2 will decompose into HS2 H transpose. And R here represents the rank of the matrix, or In other words, you can think about it as the number of clusters that you are going after. Now, in all of these cases, of course, n is significantly larger than r. The point to remember here is that h is shared between all the different data types, right? And you can start thinking about h as being a lower dimension representation of all the samples. And the idea behind non-negative matrix factorization is, again, not new. Non-negative matrix factorization has been around since 1994. And the classical representation of this problem is taking a matrix, which is m cross n, and decomposing into two different matrices. Again, r here is much, much less than the minimum of m and n. And the idea here, what separates non-negative matrix factorization from other factorization techniques? PCA is the most familiar matrix factorization technique that we can think of, but PCA does not restrict the entries in your matrices to be non-negative, right? So there is always this problem of understanding what PCA actually means. However, in terms of non-negative matrix factorization, since all the entries are non-negative, there is a benefit in terms of interpretation. So as an example, if you use non-negative matrix factorization and image processing or image identification and so on, so you can think of this as one image. And once you decompose it into its two images using, or two matrices using non-negative matrix factorization, you can clearly see here these features that become important. These are different shapes of the eyebrow, or different shapes of the mustache. So there is a interpretability associated with the idea of entries being non-negative. So let's go back to our decomposition. It is not the traditional decomposition into two matrices, but it is a trifactorization. So you are breaking each adjacency matrix into H, assay-specific S, and H transpose. Again, H is shared in all these data types. So how do we do this? So we typically use an objective function where for each assay, we are calculating how well we are decomposing, right? And we are calculating the Frobenius norm of that error in our decomposition. Now, Frobenius norm is nothing except you take the absolute value, square it, and you sum all the numbers in a particular matrix. That's essentially what Frobenius norm is. But what we are doing is we are calculating the error in our decomposition, and we are taking the Frobenius norm, and we are summing the errors in our decomposition in every layer. What we do, if we want to use this in clustering to make our life a little easier, is we add another term to this objective function, which tries to or attempts to add sparsity. So we are basically adding we are enforcing sparsity on the H matrix. And I'll talk about why that is important in a minute. So when you do typically, let's say a PCA, you will end up with representation in a lower dimensional space. And then you can think about whether you want to apply k-means or something like that. But if you just add sparsity to the objective function, what that means is instead of having to apply k-means or any other algorithm, you can take the H matrix. And for example, let's say this is an this is our n cross R matrix. And for a particular patient, so let's say we are looking at the patient which is represented in row 1, we are going to assign it to the cluster that has the maximum value. So out of these 3 columns, if the maximum value is in column 1, then the patient is assigned to column 1. And that is the benefit of just adding sparsity to the objective function. All right. Even though we can now, let's say we have that objective function and we figure out a way to essentially do this, there are still lots of issues that we need to resolve, right? Because gradient descent is basically you are starting from a particular point in the solution space and you hopefully iterate till you get to the right solution. You can get stuck in a local minimum. And that's why it's beneficial to run the solver multiple times, right? So there is a dependence on the starting condition. Then there is a question about how do we select what the optimal rank is, right? What is the optimal number of clusters? And the third is, how do we ensure that the clusters are stable, or clusters are not some consequence or artifact in how we are doing things? And the way we do this is by using resampling to our advantage. So the idea is, let's take an example where we have two different data types, and we have now converted them into similarity networks, and then we have done the decomposition for R is equal to 2, right? And instead of taking the complete data set and doing this decomposition, we are going to take 80% of the patients and do this decomposition. And we are going to then again take 80% of this data and again do decomposition. And we've done this because we do this multiple times, we can generate what's called a consensus matrix. So if we ran it 20 times, how many times did a pair of patients go together, right? So that is represented in a consensus matrix. And besides doing it for rank or number of clusters is equal to 2, we can do this for 2, 3, 4, 5, up to a specified number. Now, typically, if you really want this information to be used in the clinic, you want to limit what n is. You want to keep it below 15 or 10 or something like that. But once you have done this, you now have a cluster, a consensus matrix for each rank. And now you can use that to your advantage. So you can use things like, what are the proportion of ambiguously clustered pairs, right? So we call it PAC, and that's again not a novel idea. It's been around for a couple of years now. But you can essentially use proportion of ambiguously clustered pairs to assign some sort of measure of stability and some sort of measure of your confidence in whether your how well did you do in clustering patients in that number of clusters. And based on those metrics, such as proportion of ambiguously clustered pairs, you can choose the optimal rank. And if you choose the optimal rank, again, based on subsampling, you can assign some sort of measure of confidence in your metric, which is the proportion of ambiguously. So all of the steps that I talked about are implemented in what we call SUMO, which is subtyping using multi-omic data. And this particular tool can be downloaded from PIP. It's available on GitHub. You can use a Docker container if that's what you figure out. That's how you essentially install or use methods. And a lot of the ideas that I talked about now are embedded in what we call modes of SUMO. So prepare essentially is taking particular feature matrix or gene expression or methylation or assay and converting it to the similarity network. The run mode takes those similarity networks and runs the factorization. Interpret allows you to then calculate the importance of all of the features in determination of each one of those clusters. And evaluate allows you to compare your clustering results to some other classification. So for example, if you were doing a simulation, you can now compare the classifications you get based on SUMO to the classifications that are the ground truth that you know of. So let's test this on simulated data. So the way we did this was we started out with some ground truth. So we had two different clusters, right, in our data. Now we injected noise into this data set. So on one layer, so we created two different layers. One layer, we injected a modest amount of noise. And in the other layer, we kept on increasing the noise, right? And then we applied several methods in order to determine how well they do. And the idea here is on the y-axis, what you have is an adjacent rand index, adjusted rand index, which is a measure of how well did your clustering do in comparison to the ground truth. And as you can see on the x-axis, you have increasing amounts of noise, essentially at the standard deviation of the noise that you have. And most methods, like some, for example, pins plus, do really well up to a certain level. But when the noise goes beyond a certain threshold, you can see a precipitous fall in their performance. Some of them show a gradual decrease in performance, and you can see SUMO here also shows a gradual decrease in performance, but in general, we are able to recover the multi. So what you have here, the dots essentially represent the median performance, and the error bar or error ribbon that you see here represents the normal value, the minimum and the maximum value, because we repeat this experiment about 10 times, if I remember correctly, and the minimum and the maximum values are shown in here. So SUMO tends to do well in this simulated data set. Another thing SUMO does really well is handling missing data. So a big problem with a lot of the current methods is they cannot tolerate missing data types. So what you have to do is essentially figure out a way to impute data before you can apply those methods. And you can imagine looking at gene expression and thinking about how computationally expensive it can be to impute the data sets. Also, the performance of your subsequent downstream applications might depend on the imputation method that you choose. But what SUMO allows us to do is to essentially handle missing data by introducing or manipulating the objective function so that we do not include patient or pairwise entries where we do not have a value. There is one more tool that does missing data, and that's Multi-Omic. And what we can show here is it has increased the number of or the percent missing samples. Both SUMO and NEMO seem to be able to handle it till a certain point, but then again, NEMO seems to, its performance seems to be much faster than that. SUMO is able to recover brightness most of the time.
Any discussion?
No, Bruce.
Sorry, can you hear me?
Yes, I think there was some interruption because someone didn't mute themselves, but hopefully that's fixed now.
Yeah. All right. So as I show here, NEMO is another tool that can handle missing data. And then so in SUMO, but SUMO seems to have a better performance even when a large amount of the data is actually removed. I mean, there are caveats to this, right? If you remove so many samples that the structure in the data is lost, then none of the methods are going to work. So that is simulations, right? So let's look at how these methods perform if you apply them to real data. So we took a benchmark that was published in 2018, which is essentially 10 different cancers. And for each cancer, you have gene expression, you have methylation, and you have microRNAs. And the idea there is that we've applied all these, I think, 8 tools in order to determine how well they do in that benchmark. Now, again, as with everything, there are caveats. What do we mean by how well they do in a particular benchmark? The measure that we want to accomplish here is, can you identify at least one cluster or group of patients that shows a differential prognosis, or that is very different from everybody else, right? On the x-axis, what you see here is the negative log of the p-value from the log rank test. And that is basically, if you are to the right of this threshold, you have actually identified a group of patients that have a significantly different survival. Okay? And on the y-axis, what you have is enriched clinical parameters. So ideally, the best methods would be up here somewhere, right? And as you can see, that's not entirely true. Different methods do some methods tend to do better for certain data types, and that's sort of the idea. But overall, out of these 10 cancers, SUMO identifies a differentially prognostic subtype in about 7 of them. And for 8 of them, it does identify at least one enriched clinical parameter that is associated with those subtypes. And, as I pointed out, different methods tend to do differently with different sort of data types. The one thing that we were interested in was the ability, or we wanted the method to be stable, right? So let's say you run this method 10 times on the same data set. Ideally, you would want to, or you would expect to get the same answer. And that is true for some methods. So for example, NEMO always gives you the same answer. So we are looking here at glioblastoma data. So it's the same data set from the benchmark. And what we tried to do was essentially run each method 10 times and then ask, okay, so what is, do we get the same cluster every time? Do we get the same number of enriched clinical methods, clinical parameters every time? And for some methods like NEMO and SUMO, that is true. We do get the same results most of the time, as long as, again, you choose the optimal number of clusters correctly. For other methods, we see some variance, right? And for some methods, it's all over the place. So pins, for example, sometimes it seems to have these local minimize. So you get either this answer or this answer or this answer. Other methods, you see slight variation in the p-values. That could be dependent on how the p-values were calculated. But SUMO does seem to be stable, which is essential, which is an essential characteristic that you want if you're going to use this in the film. So so now we've shown its performance on simulated data. We've looked at a particular benchmark. We were interested in looking at one particular cancer in more detail. And some of this was driven by the fact that there was a large study that had already looked at lower grade gliomas. So it could provide us with some sort of, not a ground truth, but something that we could compare our results to. So lower grade gliomas are neuroepithelial tumors that originate from glial cells. The WHO classification is based on histology. So you provide grades 1 to 4, and those are, as I mentioned, based on pathology or histopathological characters. The last classification that has been done by WHO is in 2016, and it did include molecular status of some genes and regions. Actually, I should rephrase this. I just saw a paper 2 or 3 days ago which talked about a revised classification. So there is a new classification out in 2021, which actually increases the number of subtypes by quite a bit. But I'm not going to go into that particular classification. Most of the analysis that we've done so far was with the revised classification of 2016. One of the interesting things about lower grade gliomas is the standard of care still remains post-operative resection. So you basically operate and then therapy, and it remains the standard of care. An interesting molecular characteristic of lower grade gliomas is that about 80% of patients have mutations in IDH1, and 90% of the mutations are R132H. About 50% of the mutations of the patients have mutations in, when I say mutations, I mean somatic mutations or mutations acquired by the tumor. RNTP53, 40% of them have mutations in the TRX, and 20% in FHUR, which is tumor suppressor. So what does the WHO guideline say about this? So the WHO guideline looks at, so this is grade 4, so we'll ignore this for our purpose today. But essentially, the WHO guideline separates the tumors, the lower grade gliomas, based on IDH status. So if you're IDH wild type, there are certain other things that they look at, but essentially you become a diffuse astrocytoma, IDH wild type. If you're IDH mutant, then you look at ATRX loss or TP53 mutation. So that makes you a diffuse astrocytoma. Or if you have a 1p, chromosome 1p arm or a chromosome 19 on Q arm co-deletion, then you become an oligodendroglioma. The molecular analyses, the biggest molecular analysis that has been done for lower grade gliomas so far is a study in 2016, which looked at more than 1,000 patients, all grades 1 to 4. And what they did was essentially figured out integrating gene expression and methylation. They identified IDH mutations to be, again, the driver of the disease. So they partitioned the data into IDH wild type and IDH mutant. And then they did a combination of unsupervised and supervised clustering to identify 7 different clusters. 6 of them were for lower grade gliomas and grade 4 gliomas. And 1 cluster here was specific for grade 4 gliomas. And what they surmised were that out of these 6 clusters in lower grade glioma, there were 3 clusters that had really poor prognosis. So let's remember that as we move on. But the idea was, as I mentioned, they identified 6 methylation groups and 4 RNA expression groups. And then once they integrated everything, they identified about 6 clusters in lower grade glioma. All right, so this was in 2016. Since then, there is a group of medical researchers as well as clinical practitioners who have formed this consortium called Say Impact Now. And this consortium has gone on to make several suggestions to WHO and other classification bodies on what they think should be the diagnostic criteria for how central nervous system tumors should be classified. So as you can see here, they've had up to 7 updates since then. All right, so what we did was we started out with 530 patients for whom we had RNA-seq information, 530 patients for whom we had methylation data, and this is methylation measured a large number of probes, 450,000 probes, if I remember correctly. And we had 524 patients from whom we got microRNA data. So we had a total of 556 primary tumors, and we decided to use SUMO with this data set. So as I mentioned, SUMO uses a resampling method, or resampling technique. And as a consequence of that, what you get is a measure called the proportion of ambiguously clustered pairs, right? So this reflects our confidence in, so when you're looking at 2, you are essentially identifying that you're saying that less than 10% of the data is ambiguously clusters, which means using resampling, using multiple runs of our solver, we currently identifying the same pairs as either together or separate. And when you plot this, so on x-axis you have the number of clusters, on the y-axis you have the value of the metric used, you can see that there are 2 choices. You either have 2 clusters or you have 5 clusters. And when we look at the 2 clusters, you know, our first thought was maybe similar to all the other analyses, we are identifying IDH mutation as the bifurcating factor, right? That's the factor that's separating cluster 1 from cluster 2. But that was not the case. However, I'm not going to talk about those results for now. So let's focus on the best result that we have for 5 clusters. So on the right here, what you see is the Kaplan-Meier curves of survival for those 5 different clusters. So on the y-axis, you have the survival probability. On the x-axis, you have time. And what you can clearly see that this group, which we can, let's call it subtype 2 based on what we have here, shows an extremely poor prognosis compared to everybody else. Right? So the next question was, okay, so what is it? Like, what are we seeing? So what I have here is an alluvial plot. And what you have here is on the left, you have the WHO classification for the same 556 patients. On the right here, what you have are the glioma subtypes as determined by the molecular profiling. And remember, I told you there were 3 subtypes in this that were determined to have a poor prognosis. And those were classic-like, mesenchymal-like, and cGIMP blood. And what we immediately notice is that all of these 3 actually are assigned to subtype 2 glycemia. So for despite the fact that they were identified as 3 different subtypes in molecular analyses that were done separately on RNA-seq and methylation, when you integrate, they seem to be coming from the subtype as per SUMO. And you can clearly see that here, we do not identify IDH mutations as the driving factor in these subtypes. All right, so if IDH mutation is not driving these subtypes, what is, right? So what you have here are each facet represents a particular subtype, and each column represents a particular patient. And immediately, if you look here, you will realize that what is separating subtype 2 from everybody else is that seems to be extremely high aneuploidy and hypomethylation. And then the question becomes, okay, if that's the case, can we compare the expression profiles of these subtypes and see what is different between them? And the way we went about this was to use what's called an Excel. So we took, this is all bulk data. So we deconvoluted this data in order to identify enrichment of specific cell types that you would expect, because these are central nervous system tumors. So there are certain cell types that we expect to be found there. And what we found in this data was that subtype 2 is actually extremely similar to grade 4 gliomas or glioblastoma, right? So in essence, what we are trying to say here is that maybe subtype 2 may be a lower grade glioma based on histology. However, based on molecular information, we would think of them as grade 4 gliomas, and maybe a more aggressive therapy is what would be recommended. So this is based on gene expression data. So then we said, okay, so if gene expression shows this pattern, how does grade 2, subtype 2 compare to the other subtypes if you look at, for example, methylation? And the way we thought about this was, you know, most of glioblastoma typically we're thinking about astrocytes. So we thought about collecting information about astrocyte-specific elements. And we then looked at methylation around, or basically regulatory activity at these astrocyte-specific elements. And what you will see here is it's clear that subtype 2 has lower methylation when compared to the other subtypes when we focus at astrocyte-specific elements. Again, hinting at this idea that subtype 2 appears to be more similar to glioblastoma than to the other lower grade gliomas. So what about the other subtypes? So we did find some connections in terms of enrichments of these subtypes to known subtypes as per the WHO classification. And there were some interesting facets to it. For example, if you look at subtype 1, 128 of the 130 patients there have a methylated promoter for NGMT, which means that if you were prescribed them temozolomide, you are likely to have a good recovery. So promoter methylation of NGMT have been associated with success or response to temozolomide. So subtype 1 patients might respond better to that particular. Subtype 1 is also enriched for oligodendrogliomas and all these other. So as I mentioned here, we took gene expression, we took methylation, and we took microRNAs. But another important data type that people often talk about is somatic mutations, right? So what happens if we add somatic mutations to them? Surprisingly, not much changes, which tells us that one way to think about this is that your somatic mutations have the same information content that is available through methylation or gene expression or the combination. So all in all, SUMO appears to integrate data types, and you can use it for unsupervised learning. A big positive is you can allow missing data. So you can, in fact, more patients and more data types as a consequence of that. The downside of anything like SUMO is that you have now transformed your data from gene to patient. I mean, in a matrix, if you think about a patient cross gene, to now this patient information. So if you are interested in identifying which gene or methylation probe is driving these groups, then you have to additionally do some post-processing. So in SUMO, we do provide a module to calculate feature importance. And right now it uses a gradient boosting tree framework. However, there is a lot of scope, there are a lot of improvements that can be made to that particular. Now, I've told you all the great things about SUMO, right? But there are several severe limitations in how we do things. One of them is there are limitations to consensus clustering. There is a bias to sometimes identify a higher number of clusters. Besides that, it does not test for presence of structure. So we can test for 2 clusters onwards, but what if there are no clusters in that particular data? There is no test as of now in SUMO. This is particularly important because in recent reports, like for example, colorectal cancer, there have been reports talking about that there are no discrete subtypes. And in fact, you can think of colorectal cancer as being on a continuum, right? So it's difficult to essentially assign patients with specific groups. We also use resampling, as I mentioned, which can be extremely slow if you're looking at extremely large data sets. As of now, we do not consider relationships between the various subtypes, right? So for example, if you were indiscriminately just you kept on adding data types, you might come up with two data types that have redundant information, and you might either overweight the relationships in those redundant data types, or you might basically come up with a result that's not correct, right? We also have hyperparameters. So I talked about how we are converting Euclidean distance into similarity using something similar to nearest neighbors. So K is a hyperparameter. There is another hyperparameter which is of the Gaussian linear basis function called mu. And sometimes when you essentially try to train these hyperparameters, you can be careful about that. And autocorrect definitely decided multi-omics is not a word. So multi-comics apparently is a word. But there are limitations of multi-omics clustering in general. For example, what if the clustering based on gene expression outperforms the clustering in combined coordinates? So those are some of the limitations. That being said, SUMO, I think, is a right step forward. There are a lot of improvements that can be done. And I would encourage everybody to try out SUMO, and you can install it using Pip. We also have a Docker container. Install it that way. And in the end, of course, I would like to acknowledge all the people who actually did most of the work here. Carolina, Ajay, Nathan, John, Virginia, and Jamie and Lucian. Supported that. So, all right, I'll take some questions, but again, this is a GitHub repository. Please feel free to try installing it using it on your data sets. You can install it through Pip or use a Docker container. Squash bugs. You can essentially suggest improvements, and I'll take any questions. Thank you.
Thank you very much for a great presentation. I have a first question from our audience. So I'm going to read it now. Lyubov is asking, are there integration methods that consider relationships between different data types? If yes, did you compare them with SUMO? No. So as far as I know, I haven't seen any method that does that. So you have to understand, like, there is a general machine learning community, and then there is a bioinformatics, people who develop machine learning methods for biological data sets. And in the general machine learning community, I have seen a few methods which tend to do something like that. They use, they basically compare, I think it's called Shannon's entropy. They compare the data types in terms of the structure. And then if two data types have structures that are extremely similar, you can only choose one of the data types for integration. But no, I have not seen any methods doing that in the bioinformatics space. And that is something that we want to do. So we are looking to improve some of that. But that's a good question. Okay, so while we are waiting for more questions, I'm going to ask mine. You mentioned, so my question is maybe about future development. So is there a possibility to, when you have a new patient coming in and you have matching data types that you used previously to cluster the patients, can you add that to your data set and kind of impose it on the clustering that you've already made to cluster it to some of the clusters that you already identified in the data? Yeah, so there are two ways of doing that. Currently, as I said, we can identify the genes or the features that are driving each cluster. And then if a new patient comes in, we can restrict our analysis to a classifier that's built on those data, on those features. The second method is to essentially do what's called a diffusion
on the patient similarity network, right? So add this new patient to the patient similarity network, and then ask how far is this patient from each of the groups, and assign it to the group that's the most similar. But that is again, that is a disadvantage of this approach, is that that sort of an analysis is not trivial. However, I think once you have established the subtypes, what you want to do is identify biomarkers for those subtypes, and then restrict your assignment of future patients to a classifier that's made from those biomarkers.
Thank you. I have another question. So you have mentioned, that is a question that I got in my secret chat. So I'm not going to reveal my source, but the question is, you have mentioned that it is possible to find the features that differentiate the clusters with SUMO interpret. However, the factorization is run on similarity matrices and not on feature matrices directly. So how these features are determined?
So currently they're determined using a gradient boosting framework, but you can think of you can use anything, right? You can think of, it becomes a very general machine learning task where you have a high dimensional data set and you are trying to identify whether a particular feature is good in discriminating the groups that you've identified, right? So it becomes like a classification or a feature importance problem. The current implementation is a simple gradient boosting network. So it's like similar to random forest, but it uses a larger number of trees and also incorporates weakly separating trees. So it's an ensemble training network based on tree. And I can also answer this question another way. This is how I would like it to be done. Is essentially you take each feature and you build a patient similarity network on that particular feature. And then you ask the question, if I had this feature and I use the same number of clusters as my final answer, would I recover the relationship for this feature as, is the relationship between the cluster of the patients same now as it is for the final answer? So you can use things like purity or adjusted rand index or something like that. And the higher the adjusted rand index, the more that feature can accurately discriminate to the existing answer.
Yeah, makes sense. So I think my last question would be, you mentioned it a little bit. So how do you deal with the fact that somatic mutation data is so much different than expression and methylation? And how do you deal with that when you combine it with the other data types?
Right. So all the other data types, as you rightly pointed out, are continuous, whereas somatic mutations are discrete, right? So there are multiple ways you can deal with that. The way we think about them currently is we think about them as acting on a protein-protein interaction network. So we take a protein-protein interaction, a tissue-specific protein-protein interaction network, and then use diffusion. So essentially think of our mutations as having certain weight or heat, and then the protein-protein interaction network will diffusing that heat, right? And that transforms your discrete matrix into a continuous matrix, where the expectation is that the genes with the somatic mutations still have high values, but the other genes also get multiple values, and those values are dependent on the connectivity in the protein-protein interaction network. And then once we get that, we use that as input to SUMO. But that's a great question.
Thank you. That's a really cool method. I don't see any more questions, and I think we're a little bit over time. So let me just thank you for an amazing presentation, and thank you for joining us. And if any more people have any questions, you can either post them on GitHub or via email, or ask our Aakrosh in one of your meetings with the mentor. Thank you very much for a lovely presentation.
Thank you.